So next we have Dr. Irina Shchukina coming from Washington University in St. Louis, who's going to talk about the non-specific cytotoxicity of memory CD8 T cells, which drives excessive damage in old tissues. Looking forward to hearing your talk, and thanks so much for being here.
Hey, thanks you for inviting me. I'm going to talk about one of the projects we have in the lab that focuses on how aging affects the immune system. And previously, we pretty comprehensively characterized compositional changes between different compartments and mice, and we could identify how populational dynamics changes between young and old mice. However, the next step was trying to assess whether or not there are any functional change between different population of CD8 T cells present in young and old animals. And when you want to do the simplest experiment about CD8 T cell functionality, you usually just sort population that you know are more or less homogeneous, and we would do it following our single cell data. So this is our gating strategy. We would have memory naive CD8 T cells sorted from young mice, and from old spleen, we would sort naive cells and also like two distinct memory populations based on PD-1 expression. And the simplest thing you can do at this stage is to just do ex vivo activation with anti-CD3, anti-CD28 beads and try to see what the cells do in response. And at this point, we started facing an issue because our old memory cells had significantly lower viability than young controls we plan to compare them against. And we tried to optimize it for quite some time, and nothing was really working. And they kept dying, basically prohibiting us interpreting any downstream data. And after a lot of brainstorming, we had a kind of crazy idea that we're mixing a bunch of CD8 T cells together that are the major cytotoxic cell type. What if they're not just dying cell intrinsically? What are they actually somehow cytotoxic to each other? And to test this, we set up a very basic killing assay. So we would have control cells, target cells that we know survive well in culture, for example, regular young CD8 T cells. And we would culture them alone to have some kind of expected viability in this experiment set as 100%. And then we would co-culture them with any effector cells we want to study. So we would mix them at different ratios, and we would want to see if this co-culture will lead to decrease in viability of target cells. Because if it does happen, we could interpret it as if effector cells are cytotoxic towards their target, and that we can quantify the cytotoxicity level. So following this design, we sorted our different populations of CD8 T cells from young and old mice, and it's all spleen here. And we were able to see that surprisingly, there were like only one population, which was PD-1 negative old memory cells that were highly cytotoxic against young CD8 T cells in this case. So in young mice, no cells show any activity, which is kind of what you would expect because there is like no, like these are polyclonal cells. There is like no antigen introduced. They should not really do anything besides maybe some cytokine production and proliferation. But these guys were like they were doing something very bad to their targets. And this was only happening upon activation. To test if it was just like one system artifact, we did the same similar design experiment, but with different readout and against 3T3 fibroblasts. And we were surprised to see a very drastic phenotype here as well, because you can appreciate that after 2 days together in culture with old memory CD8 T cells, they basically killed majority of these fibroblasts. There is like almost nothing is happening, and it's like highly significant compared to young CD8 T cells. So we started reevaluating all we knew about how cytotoxicity works in CD8 T cells. And we know that normally cells select their target and form the synapse to make sure that the killing is very targeted. And we tested different components of the synapse if they're required or not. And treatment with dasatinib basically allowed us to confirm again that TCR signaling is still required in this model. So like you lose the cytotoxicity if you block, and it blocks LCK pathway and everything downstream, basically. And blocking LFA1, which is a major structural component of the immune synapse, allowed us to show that, like, first of all, synapse is still required, and it's also cell-to-cell, like cell-to-cell contact is required here as well. It's not just TNF or anything soluble that drives this effect. So the major question then was if MHC1 is required. And we did it by two different models, and I'm just going to explain this part. We were using target cells isolated from MHC1 triple knockout animals. So these cells would not have any MHC or MHC1-like molecules on their surfaces. And we could show that they were killed just as effectively as their wild-type controls, which led us to conclude that this is completely MHC1 independent process. And that's in contrast to like what we normally know about CD8 T cells, because like normal CD8 T cells, it requires T cell activation, for example, in the context of infectious model. And then it goes into tissues and it selects and kills only infected cells or like only cancer cells by selecting only ones that they recognize by MHC peptide TCR interaction. However, what we saw in these old memory CD8 T cells that after TCR activation, they lose specificity and they start killing in antigen-independent manner, which led us to hypothesize that it would mean for what it would mean for old mice is that upon CD8 T cell activation, they would cause way more damage than in the young mice need to survive. And to test it in vivo, we use the model of sterile inflammation. So again, we do not introduce any specific antigen. It's all very non-specific. And concavalin A is just activates CD8 non-specifically. It's a mitogen. And it's normally studied in the context of immune-mediated liver injury. And in young mice, it requires pretty high dose, which we were able to titrate down to the level that young mouse doesn't really respond to. So you can appreciate that ILT, which is a marker of acute liver damage that you can measure in plasma, is still very low in young mice. However, it's dramatically increases in old group. And we showed then that it's dependent on lymphocytes because all drug knockout are completely protected from this. So it's not just frailty of old mice or some other issues with their livers, it's specific to the immune system and adaptive immune system. And finally, we showed the role in CD8 T cells here because if we deplete them, we lose significantly lose this effect. And we can see these not only in liver, we also see the same pattern in kidneys and other organs. So we can conclude that it's like systemic issues. Again, it's not organ dependent, they are everywhere. So the next question was how these cells kill. And we actually done a bunch of profiling of these cells, and they, based on flow standard like markers, they look completely normal. They are not more effectors or more activated. However, the transcriptional signature is very, very different. And cytotoxic pathway is the most upregulated pathway in these cells. So we went ahead, like all machinery is upregulated, and we got kind of lucky because we went ahead and tested Fas ligand first because there is a very good blocking antibody available. So we did this, and we were able to show that this treatment with blocking a Fas ligand interaction completely abolished our cytotoxicity in both of our models. So we do know that this is completely Fas ligand dependent mechanism. And finally, we wanted to see if we can figure out why this is happening and what's upstream of the phenotype that we see. And we used the tool that our lab developed that allowed to mine publicly available bulk RNA-seq for the signature of your interest. So basically, I used transcriptional signature that I had, and I was able to find the paper that published previously RNA-seq that has a very high enrichment with mine. And this was the RNA-seq that compared CD8 T cells exposed to radiation-induced lymphopenic environment to, comparing to wild types. And since transcriptional signatures matched very, very well, we assumed that maybe the function going to be also similar. So we just reproduced the experiment that was published, and luckily it did reproduce and it worked very, very nicely. So I would irradiate young mice, and it's all done in young mice completely, regular wild types. I would irradiate them the day before, then I'll transfer normal young CD8 T cells in these irradiated animals. I will wait for 20 days and sort them out for a killing assay. And we were able to show that this is a completely similar non-specific cytotoxicity exhibited by these cells, and it's also Fas ligand mediated. And since these are young animals, we were finally able to like play around a little bit with genetically modified mice. So we were able, for example, to show that if I do the same with OT1 RAC knockout donor cells, they still kill, they even kill a little bit better than the wild types. So it's the whole process is completely antigen independent, both acquisition of the phenotype and the killing. So just to summarize, I wanted to try to place it in how we see it in a big picture. And I think everyone knows that old people and all organisms have way more issues with different infections and mortality is significantly higher. But like why exactly, it's very hard to pinpoint. And mostly because we think that it's a lot of different things that are going on at the same time. And what we think is that probably this loss of specificity and downstream excessive tissue damage post-activation is probably what drives at least like part of these susceptibilities. And we are very excited to move on with the study and first of all try to see if Fas ligand blockade will be able to reverse this phenotype and improve the survival in like different inflammatory conditions. And second, since we now know that lymphopenia is another context in which this is relevant, we want to see what other contexts these phenotypes is consistent with and where it arises as well, and if we can test anything in there as well. With that, I would just want to thank you, my lab, and everyone who helped with the process, and I'll be happy to take the questions.
Amazing. Thank you so much for that fantastic talk, Dr. Shchukina. Just a reminder for everyone, if you have any questions, please put them in the Q&A or feel free to put them in the chat as well. We definitely have time for questions with our speakers, so feel free to take this opportunity to ask them questions. I will start with a question from Dr. Fish that's coming from the chat, who is asking about which infection systems maybe you think about employing within this model. If you have any thoughts on that one right now.
So it was actually published by one of other groups in a little bit different context that this treatment worked in mouse model of COVID. We are also currently working on flu because it's one that has a very strong aging phenotype. And it's also like we do know that there is like way more tissue damage in old mice and we're trying, but it's hard because we need to time, like we want to not compromise T cell response too much, but prevent tissue damage. So it's a little bit, it's not as straightforward as it could be, unfortunately.
No, it never is, but no,
Yeah.
absolutely. Here trying to strike that very fine balance. Other questions as well coming in. I guess I'm also just going to take an opportunity to ask about more of the apoptotic methods as well. So you're definitely taking a deep dive into the Fas and Fas ligand pathway, but do you think about kind of exploring other pathways and within this T cell response, or do you see it kind of mostly within that avenue there with Fas?
We haven't explicitly tested it, but it looks like since it's completely like it blocked to zero. So I don't think like addition anything on top of it will actually change. And I think this is the most interesting like potentially therapeutic window because in infectious model they use granzyme and like Fas interchangeably. So like for them to lose function, you need to knock out both. So the idea here is that if we block Fas, they will still have granzyme be perforant arm to actually function for specific killing. So the hope is that it's completely Fas ligand dependent. Yeah.
Very interesting. Question from Payam as well, who's asking if you can comment on the immunometabolic fitness of the age CD8 population.
So this specific one seems more or less normal. So they like besides this like weird phenotype, they survive pretty well. There is like this another PD-1 high population that's significantly more, they like more terminal differentiated, so they like in completely different state. But these ones, and also like the ones that we see from lymphopenia, they actually like do not, they still like on RNA-seq, for example, they still cluster together with regular memory cells. It's a transcriptional shift, not like different state completely. So we think they're like, we don't see any evidence that they are abnormal.
For sure, for sure. And I guess just a final question. This
Hmm.
was a great comment on the age-related features of the CD8 T cells, but do you see any sex-related differences within this pathway?
We use both male and female mice in this project, and we never seen any discrepancies. I haven't tried comparing like how cytotoxic they are. There might be difference. We do see sex-specific differences in some of the aspects of CD8 T cells, so I wouldn't be surprised. But we do think it's happening in both. In which it's more severe, we haven't looked yet. Hmm.
Gotcha, gotcha. I think
Hmm.
one last question. Let me just take a look at this. So this is coming from Emily. So with the effect of lymphopenia, do you think the signature is
Hmm.
driven by the absence of other immune cells or signals like extracellular ATP that increase after the cell death caused by irradiation? Great question.
I think that this is driven by whatever, like we know that it's, for example, IL-15 dependent. Like I haven't time to show this data, but we think it's like more of like homeostatic proliferation, and it connects with aging as well because you don't have like functional thymus anymore. So to maintain the numbers of CD8 T cells, they just have to proliferate more. And we know that it's probably just availability of like it's kind of loss of other cells and thus the need for to proliferate to fill the niche and availability of factors.
Gotcha. Got
Hmm.
Amazing. All
Hmm.
right. Well, I think that is all for our questions today. Thank you so much, Dr. Shchukina, for coming
Thank
and
you.
sharing your work with us. We really appreciate it.